system. Uh, Complement system. This is a part of innate immunity consisting of plasma proteins that help antibodies and phagocytes to clear microbes. There are approximately 30 proteins mainly produced by the liver circulating in inactive forms. The complement system is a part of your immune system that defends your body against injury and foreign invaders like bacteria and viruses that make you sick. Your complement system activates proteins that work with your immune system to keep you healthy. Group of plasma proteins, complement proteins produced in liver helps to destroy pathogens. Functions of complement system. Opsonization. Major molecule C3b coats microbes to enhance phagocytosis. Second is cell lysis, creates spores to form two microbe dyes, forms membrane attack complex MAC C5b to C9. Third is inflammation, C5a, strongest chemotactic factor, small fragment C3a, C4a, C5a stimulate inflammation. And fourth is clear immune complexes, removes antigen antibody complexes to prevent tissue damage. Complement activation pathways. There are three pathways, but all lead to C3 activation, formation of C5 to C9 MAC, and destruction of microbes. Classical pathway, alternate pathway, and lectin pathway. First is classical pathway. Antigen antibody complex requires IgG or IgGM steps. C1 binds to AB on antigen, splits C4 to C4b, C2 to C2a, forms C3 convertase is equal to C4b to A. Fourth point is C3 converts to C3a inflammation plus C3b opsonin. C4 to A3b is equal to C5 convertase to enters terminal pathway. And second is alternate pathway. Direct activation by microbial surfaces, no antibody required. Spontaneous C3 hydrolysis, C3 converts to C3b, capital B, small b. Lectin pathway. Third is mannose binding lectin, MBL binding to microbial sugars. MBL activates MASPS. C3 converts to C4b2A, same as classical pathway. Terminal pathway. All pathways generate C5 convertase to C5b. C5b joins C6, C7, C8, C9 to form MAC C5b9. Punches hole to create cell lysis. Regulation of complement to prevent self damage. C1 inhibitor C1 to INH DAF decay accelerating factor oblique CD55. CD59 blocks MAC on self cells. Factor H and I regulate C3b breakdown. Biological functions of complement. Complement proteins perform four major biological roles that protect the body from infection. Opsonization of microbes. First, opsonization of microbes. C3b and C4b coat the microbe, coat the pathogen surface, enhances recognition and ingestion by phagocytes, macrophages, and neutrophils to key receptor CRI on phagocytes. C3b is equal to better phagocytosis. And second is chemotaxis and inflammation. So small fragment C3a, C4a, and C5a acts as anaphylotoxins. They increase vascular permeability, promote degranulation of mast cells, basophils, and histamine release, recruit neutrophils and other immune cells. C5a is the most potent chemotactic factor. Third is cell lysis by MAC. Complement protein C5b, C6 and C7, C8 and C9 assemble to form the membrane attack complex called as MAC. MAC insert pores into membranes of gram-negative bacteria, invade viruses from pathogen, causes osmotic lysis to form pathogen death. Fourth is clearance of immune complexes. Complement binds antigen antibody complexes, help transport them to spleen and liver. C3b converts immune complex deposition in tissues. Protects against conditions like glomerulonephritis, enhances B cell activation via CP2. CR2 removes apoptotic dead cells, helps shape adaptive immunity. Summary: opsonization C3b, inflammation C3a, C5b, chemotaxis C5a, cell lysis through MAC C5b to C9, immune complex clearance. Complement deficiencies. This occurs when one or more complement proteins are missing or non-functional. They lead to recurrent infection, autoimmune disease and inflammatory disorders. C1, C4, C2 SLE-like disease. C3 deficiencies, severe recurrent infections. C5 to C9 deficiency, Neisseria infections. C1INH defect, hereditary angioedema. DAF CD59 defect, paroxysmal nocturnal hemoglobinuria.